Hello everyone. Achieving senior academic promotion at the University of Toronto is a major accomplishment. It shows a level of national or international reputation and excellence in research, creative professional activities, or teaching. Those achieving promotion this year demonstrate the breadth of expertise in our department and are very deserving of their promotion. Thank you to everyone on the Promotions Committee for their hard work and dedication, particularly my co-chair Sharon Nofek-Moses and Patricia Cayetano, without whom none of this would be possible. We congratulate and celebrate all those promoted in LMP this year.
My name is Fangyi Lu, and I'm a breast pathologist at Sunnybrook Health Sciences Center, and I'm also a faculty member of the Department of Laboratory Medicine and Pathobiology at U of T since 2012. I think this promotion really means validation and recognition of all the works and efforts I put in. But I also hope that my promotion can act as a template for other faculty members who is interested in pursuing medical education as a platform for their promotion, which is sort of not a very common route in pathology.
I'm Tracy Stockley, and I'm head of clinical lab genetics at UHN. So I'm a clinical lab molecular geneticist, and my work involves making new diagnostic tests and then running the clinical service to provide that for patients. Yes, it's a great recognition of work over many years, so that feels great, and it also makes me, be grateful for the people who've helped me along the way.
I'm Michael LaFlamme. I'm currently an associate professor to be promoted to full professor at LMP at University of Toronto, and I'm a senior scientist in the McEwen Stem Cell Institute here at University Health Network. So first reaction is tremendous relief. It's a process that's well explained to you. It's transparent, there's clear instructions, but it's still a bit of an anxiety provoking process. You're going to put together a lot of information about yourself, and then anonymous people somewhere are going to read it and submit critiques. But also gratitude, gratitude for all the folks that I've worked with in my laboratory. I was first appointed to associate professor level in 2012, and I've been here at University of Toronto since 2015, and during that time I've been fortunate to work with a lot of really talented graduate students, postdocs, and staff, and any promotion I get is really to their credit.
My name is Kathy Chun, and I am one of the clinical laboratory directors here in Genome Diagnostics at SickKids. It means a lot. It's a validation of all the work that I've done. You go month to month, year to year, or whatever. You're just so engrossed in your work. It's sometimes difficult to kind of step back and say, oh, what have I done to date? But the promotion was an opportunity for me to actually take that step back and look at what I've done, and it was a significant amount, and I'm proud of what I've done, and it's been acknowledged by my peers, I think, by allowing this promotion to go through.
So my name's Phidias Diamandis. I'm a neuropathologist. I'm a scientist and educator at the University Health Network and the University of Toronto. The promotion for me is an important milestone in my career, but more importantly, it really serves as independent validation that the hard work that me and my team are putting into exploring the brain and its diseases is occurring at a high enough level that warrants distinction at the university level. So it's really nice to see that my peers, both at the university level and across the nation, that evaluated my progress, are supportive that the work we're doing is important and impactful for patients and pathology in general.
My name is Roberto Melano. I am a scientist at Public Health Ontario. I work in the laboratory. This promotion is a huge achievement because it's coming from one of the most important universities in the world, I would say. and is a huge push for my career, academic career, but also for my scientific career at PHO.
So my name is Samir Mubaraka. I'm one of the infectious diseases physicians here at Sunnybrook Health Sciences Center, and a medical microbiologist and a clinician scientist at Sunnybrook Research Institute and in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. So to me, this promotion is important in terms of the timing, given the area that I work in and the fact that it's a pandemic. It really is an opportunity to highlight or underscore the importance of my field, virology and medical microbiology and infectious diseases, because I think a lot of times in between international outbreaks and in between pandemics, the focus recedes into the background. People forget about the importance of transmissible agents and pathogens like influenza virus and SARS coronavirus 2 and a number of other pathogens.
My name is Sharon Ofik Moses. I'm a breast and gynecologic pathologist at Sunnybrook Health Sciences Center. So I think that on one level, this is a recognition by the international scientific community, but on a personal level, it's just a milestone in a very long journey. And as a milestone, it's not an end point, so it is just a point of celebration, but the work continues.
As I said, I'm a clinical lab geneticist, so that work involves making new diagnostic tests, or especially new tests for emerging technologies where, they may have just been first published in research, and we have to figure out how to actually make a safe and reliable test that we can use for any patient that meets that criteria. So that's part of my work. The other part is really about trying to figure out how to make those tests become standard of care tests in the healthcare system. So working on things like clinical guidelines, lab guidelines, quality management, and proficiency testing, those kind of higher level things, and working with funders to figure out how they can become funded standard of care tests for patients.
So I have two roles. So I do diagnostic work. I'm a cardiovascular and autopsy pathologist and enjoy that portion of my work. But about 80% of my time, I'm a researcher, and I run a lab that's focused on developing stem cell-based therapies for heart disease, specifically the damage to heart that happens after a myocardial infarction or heart attack. So we've worked out ways to efficiently guide pluripotent stem cells, a special type of stem cell, into heart muscle cells, and we've been testing those cells in a variety of animal models over the year, and think we're getting reasonably close to hopefully moving them to the clinic.
My main interest is to find tools to estimate personal risk for cancer patients, and also develop tools that will allow us to tailor treatments on a personal level. So in other words, this is looking at prognostic and predictive cancer biomarkers. And more specifically, most of my work was dedicated to pre-invasive breast cancer, a disease that is known as ductal carcinoma in situ. This type of cancer, in its pure form, it's not killing you, it's not lethal, but a subset of patients with DCIS will go on and develop invasive cancer, and invasive cancer can kill you. So currently, we are limited in our ability to identify patients that carry this risk to develop invasive cancer. So we kind of treat everybody the same. However, ideally, we will be able to identify patients that are at a higher risk and that patients that will benefit from the arsenal of current therapies that we have.
Genome Diagnostics has two main labs. One is cytogenetics and one is molecular genetics. And what we do is we do genetic testing for all types of genetic diseases, which help in diagnosis, prognosis, and in the management of these patients. And it's changed a lot over the years with advances in technology. We're able to sequence more, find more changes. We can even sequence the whole genome now. So this provides us a better understanding of the pathobiology of the disorders, and also in how to treat these patients with precision medicine. So basically we're relying on the exact genetic changes in the patient to sort of inform us on how they can be managed.
So my academic work and interests really revolve around understanding how the brain forms during in utero life, a period where many dramatic changes are occurring as the brain is wired. So we're inherently interested in some of those processes. And then later on in adult life, trying to understand why certain diseases such as cancers of the brain occur, and trying to understand the complexity of the molecular details that drive these tumors in hopes to develop better ways to diagnose and treat these devastating disorders.
So my work has sort of two main aspects. So I'm very interested in standardizing the clinical and pathology practice of breast pathology. And then so for my promotion, I was very focused on standardizing the practice for patients who were treated with neoadjuvant therapy. And then both in terms of developing a national guideline, as well as in the digital pathology and image analysis front. And then the second aspect of my work is on pathology education. So I'm very involved in pathology education at all levels, from undergraduate medical students all the way to continuing professional education of pathologists. And then I recently also took on the challenging task of being the field director for the pathologist assistant students at U of T.
My specific work is studying bacteria and how they can survive the presence of some drugs that we used in public health, that are the antimicrobials, and how bacteria evolve in presence of those drugs and can become resistant to those drugs, and how they acquire this mechanism, how they share it between bacterial communities. And that is a huge problem for public health, because we are not having a lot of antibiotics to cure diseases that are produced by these multi-drug resistant bacteria.
So our work focuses on respiratory viruses. So right now we're working on influenza virus and SARS coronavirus 2, unsurprisingly. And we really started off looking at transmission, and that's still the focus of our work now. We use a range of tools and instruments. So bioaerosols, for example, was something that we worked with quite a bit. So looking at the aerosols that people emit when they're infected and looking at characterizing those. We also got into experimental models, so looking at in vitro, so cell models, but also animal models of transmission. And then also more recently got into looking at it from a genomics perspective. And while this all started off really in the healthcare setting and looking at it from a human health perspective, because the viruses we were looking at, especially influenza virus, prior to the pandemic, we really started looking at where these viruses get transmitted. And so we started going to, for example, agricultural settings like a swine barn to try and detect and characterize influenza viruses of swine origin. And from there on, a lot of health-focused work followed. So in working with wildlife biologists and other animal health specialists, we started looking for SARS coronavirus 2 in wildlife, and that's how, through those collaborations, we were able to detect the first SARS coronavirus 2 in free-ranging wildlife here in Canada in white-tailed deer.